and then he did his postdoc from very prestigious Max Planck University, Max Planck Institute for terrestrial microbiology. Sir has published a very good number of papers in the around 95 papers in the journals of national and international repute. Sir is also visiting professor at Xinjiang University, Henan, China, and sir is serving in the editorial board of several very reputed journals published by Springer and Elsevier. Few of them like Bangladesh Veterinary Journal, Veterinary Infectious Disease, Frontiers in Terrestrial Microbiology, Microbiological Research. I saw sir's profile in the Google Scholar. In this half year, sir has published more than 10 papers in highly reputed journals. So we can just judge the sir. Today's topic is also very, very relevant, antibiotic resistance, because in today's scenario, whether it is environment, whether it is agriculture, whether it is veterinary or medical sciences, antibiotic resistance is affecting whole of our not only humanity but every aspect of life. So I just give the opportunity to sir to deliver a valuable speech, sir.
Okay, thank you very much. So I will like to share the slides, so I can go and request you to give the permission to share the slides. Okay. Can you see it?
Yeah, yes, sir.
Yes.
Okay. So first of all, I would like to express my gratitude and thanks to Honorable Vice Chancellor Professor Rao for giving me the opportunity to give a presentation here about what I was been doing. Basically, I am not that much famous as you are thinking. I'm just a sort of simple person with some basic studies and experience, nothing special. I don't know too many things, but still I will try to share a few things that I know to some extent. There is no end of learning. So basically, it's impossible for anyone to know so many things. So I will talk about antibiotic resistance. So we all know that what is resistance and what we can do and so many things. So I don't want to give any more presentation or talk about my background. But as he told, I am from veterinary microbiology from Bangladesh Agriculture University, and working as a professor in the Department of Microbiology and Hygiene. So this is about the outline of my presentation. So I will talk about antibiotics, what are the classification, and then the consumption of antibiotics, then the resistance and how we can know and detect the resistance, why we need to think or concern about antibiotic resistance, a few things about the mechanism, some of the basic findings from my lab, and also what could be the alternative way to antibiotics, and then how we can overcome or combat the antibiotics. So if there is any problem, or if you are not able to see or listening problem, you just please feel free to let me know. So this is the opportunity for me to tell about my university. So this is Bangladesh Agriculture University. This is one of the largest and oldest university in Bangladesh. It was established in 1961. And there are 6 faculties, and we provide undergraduate, MS, PhD, many different kinds of degrees. And there are about 10,000 people, including students, staff, teachers. So this is one of the biggest university in the field in Bangladesh. And this is a view of my department. This is the new extension building. So I'm working in the Department of Microbiology and Hygiene. There are currently about 22 faculty. We have around 100 MS, PhD, postgraduate students in the department. And we teach veterinary microbiology at different levels for the veterinary students and also to the students of other faculty. So now let's go to the basic talk. So we all know that what is antibiotic. So antibiotic is a substance which basically produced by microorganism and have the ability to kill or destroy the microorganism, particularly the bacteria. The one important thing is that it has to be produced by microorganism. Suppose acid, strong acid and alkali can also kill bacteria, but they are not antibiotic because they are not produced by microorganism. So it's a chemical produced by microorganism and having effect on bacteria. And we all know that Sir Alexander Fleming, he first discovered the antibiotic in 1928 accidentally from penicillium, and he was awarded 1945 the Nobel Prize. And this is the person, and his contribution a lot. He saved lots of life during the Second World War. And since his discovery, we are still getting the benefit of antibiotics. So this is the person, Sir Alexander Fleming, who actually developed the, discovered the antibiotic. And about the classification. So we can classify the antibiotics in many ways, depending on the target organ, mode of action, spectrum of activity. So there are many ways we can classify. So some of them, for example, penicillin, they inhibit the bacterial cell wall. And some antibiotics like tetracycline, they inhibit the protein, and protein synthesis could be reversible or irreversibly stopped. And there are also some enzymes, particularly those involved in the DNA RNA synthesis, could also be a target of antibiotics. So depending on action, they could be classified in many ways. And again, based on the spectrum of activity, they could be classified as narrow spectrum and broad spectrum. So narrow spectrum means only they will work either against gram-positive or gram-negative bacteria. For example, the commonly used penicillin. On the other hand, we know that some antibiotics, they can work both against gram-positive and gram-negative bacteria, and that is called broad spectrum, for example, tetracycline. Again, we can classify the antibiotics as bacteriostatic or bactericidal. So bacteriostatic means they will not kill the bacteria, but they will inhibit the growth. So the number of bacteria will not increase. Their population will remain the same, but they will not be killed. And that is called bacteriostatic. For example, tetracycline belongs to that group. And then we have bactericidal. Cidal means they will kill the bacteria, they will destroy them. So this kind of bacteria will be killed by the antibiotic, and that kind of antibiotic is known as bactericidal. For example, some of the sulfonamide, penicillin, and carbapenem groups of antibiotic. And this slide just to give you some idea of the different targets in the bacterial cell that could be affected by different types of antibiotics. For example, penicillin, they will interfere bacterial cell wall synthesis, particularly peptide. And there are quinolone compound, they will interfere the DNA synthesis because they affect the DNA guidase enzyme. Some of them will interfere the synthesis of protein. So there are many ways, many targets that could be affected by different types of antibiotic. And ultimately, it will either kill or inhibit the growth of bacteria. However, the main concern now these days is the resistance. Since it was identified in 1948, within few years, the bacteria, they started to become resistant. So they were not being able to be killed or destroyed or inhibited by the antibiotics. It is the natural phenomenon, everyone wants to survive. So bacteria has developed the strategy to overcome the activity of the antibiotics, and that is known as antibiotic resistance. And it's a huge problem. And considering this resistance in 2017, WHO, they proposed the new classification, which is known as AWARE classification. This is basically targeting the resistance. And there are three categories of antibiotics, and one of them is called excess group, another them is called watch group, and another them is called reserve group. So in total, there is 180 different types of antibiotics that have been categorized under this AWARE group of classification. And if you think about the excess group means you can have access to these antibiotics almost anywhere, and you can easily use them. You don't need to think too much because in general way, most of the normal pathogens, which are not seriously notorious or dangerous, they are in many ways very much sensitive to these antibiotics. So you can easily use them. You don't need to concern about these, and most of the bacteria may die. And there are about 48 antibiotics, including ampicillin, amoxicillin, which actually could be effective against these bacteria because there is no resistance that much, or these bacteria have very low resistance pattern. But you have to remember they are not serious pathogens. They are opportunistic or doesn't cause too much infection. But there are some antibacteria which have higher degree of resistance and they could do some more serious damage to the body. So this group are known as high risk group of bacteria. And for this bacteria, we have to use the antibiotics, which you have to be careful and watch group. Watch means you have to keep them under watch. You have to observe their activity when you use, because some of the bacteria already have developed resistance. So you can apply this antibiotic, for example, lincomycin, levofloxacin, cefoxtin. Then you have to observe the patients whether they are getting resistance or susceptible or sensitivity, because some degree of resistance has already been noticed. So unlike excess group, in this case, you have to watch the antibiotic, I mean the patient, to see the progress because there are already some resistance. And then there is another group that is called reserve group. Reserve group in the sense you don't have access to these antibiotics anywhere without any proper justification or prescription. This is the last resort of antibiotics. Only the highly pathogenic multidrug resistance bacteria, against which neither access nor watch group could be effective, you can use these antibiotics, because least resistance has been observed against these antibiotics. So these groups include 22 antibiotics, including colistin, imipenem, carbapenem, which we only use in extreme case when there is no activity of the excess or watch group. So this is the reserve, last resort. You keep them somewhere in a reserve position, in a hiding place, so that only in case of emergency, you can use this as a last resort to save the life. And in general, only 8% antibiotics belong to this reserve group, and the remaining are either excess or watch group. And antibiotic resistance is a big problem, as I told you earlier. So it's like COVID, you know, whether you rich, poor, you are a president, minister, or a simple farmer, it doesn't matter. Everybody is under the influence of antibiotic resistance. So the bacteria can make the infection to anybody and anybody can get resistance. So antibiotic resistance is a global problem all over the world. And everybody, not only human, even the animal, agriculture, everywhere you can see the resistance. And it is the selective pressure which actually influence the development of a resistance. So there are many factors which ultimately influence the bacteria to overcome the activity of the antibiotic. But the antibiotic itself is the major driving force, because when the bacteria is exposed to the antibiotic, they want to overcome. And this is how they modify themselves, ultimately become resistant. So the main selective pressure is the antibiotic itself. And currently there are tons and tons of antibiotics are being used in case of veterinary, human, agriculture. For example, if you think about the 2010 data, more than 63,000 tons of antibiotics are being used in livestock. And currently its amount is more. And in 2030, if it goes, it will be then 100,000 tons of antibiotics will be used in livestock. So to increase the or maintain the population of the animal, to keep them healthy for better food, lots of antibiotics are being used in veterinary science. And it's also same in medical science. And you can see that from this graph, uh, current position, and by 2030, the expected number of antibiotic use actually huge. And most of them are being used in China, then USA, then Brazil, and subsequently other countries. And if you think about the prescription, this is the daily defined dose of number of prescription on average per thousands of people. So in Bangladesh, it's like around, for example, 20 to 30 prescriptions are being made for antibiotics for average 1,000 people. So if you consider the total people in Bangladesh, there is thousands and thousands of everyday prescription for the antibiotics. This is just an example how we can increasing the number of use of antibiotics. And if it continues like that, within the next few years, you can see that there will already 39% increase in the antibiotic consumption, and it will increase a lot. So by 2030, 40, 50, a huge number of antibiotics will be used in case of human, in case of veterinary, in case of agriculture. So it's a big pressure for the bacteria, and ultimately they will develop the resistance. And the resistance could be many types. It could be simple or it could be complex. So in some cases, bacteria may be resistant to one antibiotics. In this case, there may be resistance to 2 or 3 groups or class of antibiotics. Then we call multidrug resistance. In few cases, we call them extensively drug resistance because they are resistant to 6, 7, 8 different class or types of antibiotics. And like pandemic, some of the antibiotic resistance bacteria are known as pan-drug resistance. Pan-drug resistance means almost any antibiotics you used against them, they will show the resistance. So there are different degree of resistance, multi-drug, extensively drug resistance, pan-drug. So this is just to give the magnitude of resistance. And the mechanism, as I told, some bacteria, the antibiotics will work in the cell wall or target, and bacteria will alter or modify their enzyme, cell, target, DNA, RNA, so that the antibiotic will not interact, it will not find the receptor. Even it goes inside the bacteria, it will be pumped out. Even within the bacterial cell, it will not find the target because bacteria has developed the strategy to overcome the target enzyme so that the antibiotic will not interact and ultimately bacteria will be resistant. So this is just an example to show that how bacteria could be resistant against the antibiotic. The main concern is that for the last 30, 40 years, almost no new antibiotics has been developed. And that is the main concern because almost all the antibiotics so far we know have already been resistant to some extent by bacteria. And there is currently no new antibiotics which could be used more effectively. And from this graph, you can see that and since 1990, virtually there was no development of new antibiotics that could be more effective. And as I told you, this is a classical example of a review paper from Jean O'Neill, where he showed that if the resistance continue, it will kill more than 10 million people in the next 20, 30 years. Currently, about 700,000 people are dying every year because of the complication of antibiotic resistance. So people are dying from accident, cancer, suppose diarrhea, dehydration, confusion, so many other causes. But in general, it is negligible compared to the antibiotic resistance because once bacteria causes the resistance, then no drug will work and ultimately it will cause a high mortality. And this is big concern because the antibiotic resistance also has effect on the SDG, the sustainable development of goal would not be achieved. And we will talk about it later. And antibiotic resistance is also a big problem for the economics of the country because the patients currently, suppose if the antibiotic costs 10 taka, then you have to buy more powerful antibiotic which will be 100 taka rupee, then you have to pay more, even it will not work. You have to keep the patient in the hospital, then you have to pay for the hospital with the patient. Some family members will stay there and there lots of money are involved. So it is a huge burden for the country, for the health sector, for the personal family, for everybody. And this is a big issue and it will ultimately interfere the sustainable developmental goal SDG of the United Nations. For example, we know that the number one SDG criteria is the no poverty, but it will not be achieved if antibiotic resistance continue because a family will suffer a lot. They will spend everything to buy the antibiotic for the treatment. Ultimately, there will be poverty. Once you have the poverty, then you cannot do other things. And similarly, if you think that zero hunger, if there is resistance, you cannot treat the animal, the poultry, livestock, they will die. So the major portion of animal protein will not be there. So ultimately, you are not able to remove the hunger. So ultimately, almost 5 out of 17 major goal of the ATC will be affected by the antibiotic resistance. So antibiotic resistance is a huge problem. In case of livestock, it causes around 11% fall in the production. So if you think about meat, milk, egg, whatever it is, they will not be so frequently found if we are not able to continue or overcome the situation. And the situation is more dangerous in the low and middle income country because we have other problems. And to overcome this problem in 2015, in the United Nations, they took a plan, and that is called Global Action Plan. The Global Action Plan for Antibiotic Resistance propose many ways how antibiotic resistance could be overcome. Now we have to know how you can detect the antibiotic resistance. So there are many ways. In a simple way, you can culture the bacteria, and then you can do the disc diffusion test. In the culture, you put some disc which have some bacteria, and if you see that the disc is not able to kill the bacteria, then you know that the bacteria is resistant. So that is called disc diffusion test. You can also do the MIC, where you do the dilation of the antibiotics or bacteria, and then you know what is the concentration of antibiotics working. And with the chart, you can see whether the antibiotic is resistant or not for particular bacteria. Then we also have the E-test and other test. And indirectly, you can detect the genes responsible for the antibiotic resistance, the phenotype, genotype, to have some idea about the resistance in the bacteria. And this is just an example. You can see that a plate with several antibiotics, and around the disc, there is a diffuse zone. That means the antibiotic is working and it has killed the bacteria. So we can use that antibiotic. But on the other hand, if you see on the top right, the ampicillin, there is growth of the bacteria just adjacent to the disc. That means this particular bacteria, this is a avian pathogenic E. coli, it is resistant to ampicillin, similarly resistance to erythromycin, resistance to tetracycline. So this is a simple way to detect whether the particular bacteria is resistant to particular antibiotic or not. Then in my lab, we also do the MIC, and it is E-test. So you have a strip which has a gradient of antibiotic concentration from the top to the bottom. You place the disc or the strip in the disc media, and then you see the zone of inhibition. Mechanism of calculation is there, and you can have some idea what is the MIC for particular antibiotic. Now I will talk about some of the findings in my lab. So there are around 8 to 9 members now in my lab. All of them are not here. Some of them are abroad. And there are many ways and many areas we are exploring. So first of all, I work on antibiotic resistance. Then I also work on vaccines, virulence, pathogenesis, food hygiene. And in case of antibiotics, we are focusing on National Action Plan of the Bangladesh. So we have a plan for the government, and we did the analysis of the National Action Plan of the government, and we found that the National Action Plan proposed by the government is very effective, and we are following that action plan so that the government can overcome the situation. And then we made a publication about the analysis results of this National Action Plan of the antibiotic resistance. And we also, in our lab, with collaboration, we are focusing on antibiotic quality with the group from Boston, because if we use the reduced quality antibiotic, it will ultimately influence the development of resistance. So we propose that we need to focus on the quality of the antibiotics. And we also published one prospective findings in the World Health Bulletin, and this is a collaborative work with Professor Jaman from Boston. And this is our recent paper. It was accepted last week. And this is the part of one of my PhD students. Here we have shown for the first time in Bangladesh that avian pathogenic E. coli is present in the poultry, particularly in the layer. And avian pathogenic E. coli causes severe economic loss. And it also has zoonotic potential. It can cause disease to the human. So people working in the farm are in the risk of getting infection from the avian pathogenic E. coli. And about 36% of the isolates, in our case, the E. coli, were found to be avian pathogenic E. coli. And surprisingly, all of them were multidrug resistant. So they were found resistance to colistin, even ampicillin, tetracycline, sulfonamide, gentamicin. So many drugs, including colistin, were found to be resistant by this group of antibiotics. And surprisingly, not only in the feces or internal organ of the poultry, even the air sample in the farm also contain avian pathogenic E. coli. So in the environment, the avian pathogenic E. coli is circulating, and that was surprising. We didn't expect that E. coli will be found to some extent in the air in the poultry shed. And we also work on fly, particularly in the fish market. And we found that there are many flies in the fish market. They are actually directly involved in the transmission of salmonella. And many of the salmonella are pathogenic, and they have zoonotic potential, and they are multidrug resistant. And actively, the fly are transmitting this salmonella. This is not something new or unique. We already know, but no one focused with the proper approach, molecular-based evidence. So we published it in Pathogen recently. And we also did the kappa analysis, knowledge, attitude, practice of the behavior of the personnels working in the fish market. And we found that they are not concerned about the antibiotic resistance. They don't think it is a big issue, and that is a big problem. We need to aware the people more about the consequence of antibiotic resistance. So this is the basic findings. About 56% houseflies were found positive for salmonella, and almost all of them were multidrug resistance, 91.2%, including tetracycline, gentamicin, and a few other antibiotics. And then we also study the antibiotic resistance in flies in hospital, dairy farm, and other environment. Because in Bangladesh, I think also in India, other parts of this region, flies are almost everywhere, particularly during the rainy season. And they travel from here to there, and actually they are carrying lots of pathogen, including multidrug resistance, in the surface, internal, and externally. And we collect the flies from different environmental settings, including hospital, canteen, restaurant, a farm. And we found that there is a huge number of antibiotic resistance E. coli, including the pathogen. For example, the resistant genes is also there. We found XTX1, XTX2, RFBP, related virulent genes in those E. coli that were isolated from the flies. And we also found the phenotype and genotype of the resistance pattern. We confirm our findings with phenotype and also by PCR. And all the isolates of the E. coli in the flies were multidrug resistance. Around 8% were Shiga toxigenic. And this is a big concern because Shiga toxigenic can cause a severe disease in many cases. And it is also important to mention that not only other antibiotics, the colistin, which we already know that is a reserve group of antibiotic, last resort. The E. coli also showed resistance against this colistin. 26% of the E. coli we isolated from the housefly were found to be resistant to colistin, and colistin is being widely used in many cases to treat But we have to think that it's not only a human, animal, it's environment. Everyone is affected. We have to take some basic measures, starting from sanitation, hygiene, biosecurity in the farm, in the hospital environment, to reduce the load of microorganism pathogen so that we can overcome. And we also need to focus on the animal. As I told you, my background is veterinary science, so I will always focus ultimately with the animal. And in fact, more than 80% of the antibiotics is used in the livestock globally. And this is a major area that we need to focus. We just simply can't focus on human, hoping that it will help to overcome the antibiotic resistance. So we need One Health approach. And One Health means here, human, animal, and agriculture environment. Everything is considered as a single unit, everything under the same umbrella. So it's a One Health, one medicine, one world. So everyone is very much related to each other. We cannot separate human from animal, animal from environment, or environment from human or animal, because there is direct, indirect linkage, a strong interaction among ourselves. So we have to focus everything, not only the human, but also we have to target the animal and also to environment, so that with the One Health approach, we can overcome the situation. More importantly, we have to use the specific treatment for the specific pathogen using the sensitivity test. And we can only do that if we are able to establish the sensitivity service everywhere. Then we can have idea which antibiotic is good. And if we use that, then it will ultimately work against the particular pathogen. And ultimately the pathogen will die and we don't need to use the other antibiotics. And there are many other alternative ways currently people are focusing. For example, in many cases, the Neem, which is a plant found in India, Bangladesh, in many cases, people are focusing on research to use Neem to inhibit the bacterial growth, to kill the bacteria to some extent. Even the garlic, ginger, different spices, we need to explore those. Already there are tons and tons of publication paper in this regard that they could have some effect on certain bacteria, but we still need to focus more so that we can find some natural ingredients that could be more effective like antibiotic to overcome the antibiotic resistance. And there is another alternative could be the bacteriophage. And this is the virus which actually affect the bacteria. So we can use the virus to kill the bacteria which is not being killed by the antibiotics. And that is called phage therapy. And there are many labs in the work currently working on the use of particular phage for a particular treatment of antibiotic bacteria. In my previous lab in Canada, when I was doing masters, they used to focus on phage for the treatment of Salmonella and E. coli infection in pig. To some extent, they were being successful in amen, but still more works need to be done to have an effective phage therapy against multidrug resistant bacteria. And this is just a simple graph. You can see that the number of bacterial load is actually decreasing once they are being exposed to the phage. That makes sense because the phage actually kill the bacteria, destroy the bacteria. Ultimately, the number will gradually decrease and to some extent, it will cure the infection. And there is another term called antibiotic stewardship. And that is actually an approach where we need to focus so many things so that we don't use antibiotics everywhere. Unnecessary use of antibiotics have to be stopped. Only effective antibiotics in a precise dose could be used to get cured. And such a big problem could only be handled if everyone take cumulative effect. As I mentioned, we need One Health approach to overcome this kind of big issue. And this is all for the moment. So thank you all. And I would like to acknowledge Professor Rao, the Honorable Vice Chancellor of Mizoram University, for giving me the chance to share some of my ideas that we are being doing. And also the authority of the Mizoram University, all the ordinance, and also the people from my university. Thank you very much. And this is all. Thank you.
Hello, Professor Tanvir ji, for your good presentation and very nice lecture. It was very nice. And so, what is your opinion about the antibiotics which are being suggested for COVID-19 control? Do you have any idea? Because I seen your review article on COVID-19. What is your idea about COVID-19? The antibiotics which are being suggested by many people, and they are having some kind of apprehensions also that they should not be used like that. What is your, can you add up some information on that COVID-19, that is antibiotics which are being suggested for that? Hello? Are you hearing me? Tanvir ji, are you hearing me? Hello?
I think interruptions for some reason, yes.
Yes, sir. Yes, sir. Sir, I just would like to know about your opinion on the antibiotics which are being suggested for COVID-19 control. There are many antibiotics are being used. Whether they are being really effective or not, because I saw your one of the review article on COVID-19.
Yes. I mean, that's a good question and a big issue. And this is actually something we need to concern. People are thinking that there are some antibiotics that are effective and should be used. But this is my personal opinion. I am not expert. But in many cases, azithromycin and doxycycline, which are being used, were found to be effective to some extent to overcome the lung infection. So maybe, I mean, I'm not the right person to answer because I'm a veterinarian, you know what I mean? A medical
Yes,
expert
sir.
will be able to give you the, but I think, yeah, yes, some antibiotic could be used with proper caution, but I'm not the expert in the field. It's something related to the medical science. So it's not wise for me to
Yes, sir. Correct.
comment.
Sir, what is this infectiousness of COVID-19 through aerial spread and also recent, actually, in the last 1 week, I have been listening through aerial spread, it is going on to person to person. So, do you have any idea about that? Because I'm asking about this because you have written a very good review.
Yes, at that time in that review, we found that, at that time there was not information about the aerosol spread. But now these days, like you, I also noticed the report and people are thinking that yes, it do spread by aerosol, but probably we need further study. But
Yes, sir.
as like you, I have noticed that people are
Yes,
reporting
sir.
that it's aerosol as well, including the fecal transmission is there also to
Oh,
some extent.
okay. Sir, what about the possibility spreading through? Because the other questions, participants' questions, Sanjay will attend. Before that, I just wanted to request my 2, 3 questions. So, what about the eating through animal meat? Is there any possibility that COVID has a possibility to spread?
Yeah, that is also under investigation. There are some reports, you know, that like in zoo in UK or some other place, they found that they isolated detected the virus in the animal, but there is no concrete proof that it's could be transmitted through the animal product, as far I am concerned. More study need to be done
Yes, sir.
to confirm this thing.
Yes, sir. Yes, sir. Sir, thank you, sir. I'm done with my doubts, and I think that I will hand out to Dr. Sanjay to ask the questions raised by the participants, sir, because there are some participants' questions, sir.
Okay, sir. I will attend the participant question. So, sir, thank you very much for very enlightening lecture in a very simple and very liquid manner, which could be understandable by any of the layman persons. Sir. Very good presentation, sir. So, sir, some of the question which is being asked by the participant. So, one of the question is the sir, can antibiotic resistance be reversed?
Yes, of course it could be reversed. So as I told you, it's the selective pressure. So if we stop the use of antibiotic for a long time, bacteria will ultimately take out the activities that was being responsible for the resistance. So ultimately it will reverse. Yes, of course, that's a good question. Yes. And this is why you are saying we have to stop indiscriminate use of antibiotic. Because if we stop it, then it will be sensitive. Bacteria will become sensitive. Resistance will go. Yes,
So,
of course,
yeah,
it
yeah.
will go away.
Thank you, sir. Next question, sir. Can probiotic reduce antibiotic resistance?
Probiotics, that is a different case because probiotics actually it mechanism is different. It stop the site where other pathogen can bind, so that the other pathogen is not able to not able to produce the disease. So probiotics actually, to my understanding, it doesn't have direct effect to reduce the antibiotic resistance, but it can stop the antibiotic resistance bacteria to establish infection into the body to cause disease. So indirectly, it is actually eliminating the pathogen or multidrug resistant bacteria to cause disease. On the other hand, in many cases, people are concerned whether the probiotic itself could be involved in transmitting resistance genes horizontally to sensitive pathogen. Some issue is there. Okay.
Okay, sir. Another question, sir. Why not so much of antibiotic developed after 1990? In your presentation, also you make this point. So, what is the main reason, according to you, sir?
Main reason actually people are targeting, but you know there are some class of compounds that has the antibacterial activity. So far, because of the science, we already have explored all of them. So we are not able to find any more new antibiotics. Probably it is out there somewhere. We need to screen more samples. And someone from pharmacological background will be able to explain it more clearly. But to my understanding, we are not exploring a lot. And what we are supposed to work is already been identified. We need to put more intensive focus. That could be one of the causes. We are not able to find resistance.
So, sir, another question is that how bacteriocins are said to be effective against antibiotic resistance?
Okay, bacteriocin is a substance which is produced by bacteria, which has activity on other bacteria. So it doesn't work like antibiotic, but it will interfere the growth of the related bacteria. Ultimately, if that particular bacteria is resistant, it will not be able to produce disease. So indirectly, it is working against antibiotic resistance. Indirect way, but bacteriocin is a different compound, which is produced by bacteria, has inhibitory effect on other related bacteria.
Sir, another question that especially in the Bangladesh and India, where waterborne diseases are very common, and antibiotic like tinidazole, ornadazole work for disease like an inflammatory bowel syndrome or Crohn's disease. And if yes, should the treatment be short or long?
That is, I think we have to maintain the dose prescribed, and I'm not sure whether it should be long or short, but if we make it longer, probably it will give more chance for the bacteria to develop resistance. Better to keep it for a short time, my personal opinion, but maintaining the dose, that is the main concern. We have to think about the MIC and the recommended dose.
Another question is that how to increase the immunity, it is a very general question, immunity of our body against antibiotic resistance. How we can...
Okay, so this is a general question. Suppose you need the physical exercise, keep yourself free from any anxiety, and then have good food, good company, vitamin C, some of the spices, friendly environment. Do not think about something bad. So these are general ways how we can improve or boost our immunity. Be healthy. Try to be healthy with the basic approach without chemical.
Another question is related to COVID spread, and Ayush government of India, it is a ministry that they are using arsenicum album. So the one participant is asking whether it is beneficial for suppressing COVID. So what is your view as an expert that this medicine, homeopathic medicine being prescribed, preventive medicine?
I mean, as a scientist, we need evidence,
Yes,
without doing proper study, you have to use the drug arsenic compound and then see whether the patients are responding or not. We cannot say anything, but I also noticed even in Bangladesh, people are suggesting to take that kind of that kind of homeopathic medicine. Probably in cases it may work, but what is the underlying mechanism and what actually the basic, we need to study, defined study. Then only then you can say exactly whether it's working or not.
Yes. Sir, last question that in the Bangladesh, recently the doctors are prescribing ivermectin in combination with doxycycline for the COVID. So how, as expert, how you think that it is a better combination, like other drugs like remdesivir, Fevipar, they are already there. So, but if these cheaper drugs which could be prescribed, what is your view on these, this ivermectin and doxycycline against COVID?
I mean, I think in many cases, people are a bit confused. I am a veterinary microbiologist. First of all, you have to remember, I'm not a medical doctor. So I'm not in a position to give that kind of answer. Although I have my opinion, but it's just my personal opinion. But as I told you, I'm not a medical doctor, not an expert in COVID. So in many cases, my understanding is just like you. I don't know exactly what is the mechanism, but I hear that To some extent, probably they interfere the DNA, RNA synthesis, and somehow they stop the virus replication. But it is just like you. I also have that kind of question, but I am not the actual person to answer COVID to that extent. As I told you, I work more on veterinary science, veterinary microbiology. Yes, I don't know actually what is the underlying mechanism or whether it's working or not. But in many cases, many things are working, but we don't know what is the cause. Maybe we need to explore more.
Yes, sir. One of my personal questions, sir, from my side, that in many cases I read that people are now prescribing, trying to use essential oil, which is derived from plant, against these antibiotic resistance bacteria. People are some people are working that how this essential oil could be. So because you are telling in the last there could be alternative therapy like using the medicinal plants, even the bacteriophage. So these whether essential oil could be really effective against this antimicrobial resistance bacteria.
I mean, maybe they may have some effect, but personally, I don't know because I don't have any experience or I didn't came across any literature suggesting that yes, they have used it and found this and this and this. So as I told you, there are so many things that could be effective against antibiotic resistant bacteria. We just need to explore more and need more scientific way of research to know exactly whether it's working or not and what is the mechanism.
Yes.
It's difficult to say for me.
So once again, sir, very much thank you for attending all the question in a very appropriate manner, because until unless we find the scientific basis, we cannot.
Yes.
Yes. So that's why
yes,
we need to have a proper scientific study. But in the
exactly.
COVID case, we are trying all over the world. We are trying whatever we can try, but until unless it is scientifically proved, it will come along. So thank you very much, sir, for very valuable lecture. So some of the
okay. Thank you very much. Thank you all.
So
okay. Thank you all.
So
okay, thank you
just I will propose the vote of thanks on the behalf of Mizoram University. So Honorable Vice Chancellor, sir, respected Professor Rahman, sir, our most valued speaker for the session, and all our valued participants, ladies and gentlemen. So it is a privilege to propose the vote of thanks on this occasion. So I sincerely thank to Honorable Vice Chancellor for giving us opportunity to organizing such a valuable webinar on a very genuine topic. I can say very genuine topic. Very means it is a problem which is just it can be a buzzword in the media and everywhere, because it is likely to affect the people and likely to affect the sustainable development goals which are proposed by United Nations, especially in the developing country, poor country, where the purchasing power party for the medicine is very low. So if you will not target such issue, because when we last year also one of my colleagues wife admitted to hospital, and just sir is telling that they are pumping antibiotic like anything. So it could affect anybody. It could be affect anybody. So
yes.
So very sir, I sincerely thank you, sir, for giving a very valuable lecture. So I take this opportunity to sincerely thank to all the participants, resource person, ICT centers who organize all the technical expertise, especially Mr. Suraj and system administrator Mr. Tulanga. And I also sincerely thank to our controller of exam. In spite of his very busy schedule, sir has attended, try to come to the schedule and try to interact with us. So overall, I thank everybody. Thank you very much, sir.
Thank you very much. Thank you very much.